Yes, I thought I would just spend a little short time talking about sort of getting the stage the set for our Alpha Clinic doctors. Like, what do we do? I'm at the Sanford Stem Cell Clinical Center director, but we also have the Alpha Clinic within the clinical center. And here's our whole team here at our outpatient KOP space, having for our picture, because it takes a whole team to pull this off. Really, we're the sort of clinical trial unit for the Institute. And as Dr. Jamieson alluded to, we've been active for a number of years, so we've kind of matured into a full service operation. And I just wanted to highlight some of the operations that we do. I think the biggest thing that we do is run these clinical trials in regenerative medicine that includes the cell, gene, and stem cell targeted therapies. But we also have both an inpatient and outpatient unit. We are doing now standard care treatments such as TILS therapies and introducing, as these new technologies get approved, the idea is that we'd be also applying those both in the inpatient, outpatient setting. We also have a START team, and our START team is out there having pamphlets. So they provide assistance with interactions with the FDA, how to get investigators coming from that bench to bedside, getting to an IND stage, helping particularly with some of the new CERM grants as they have changed, and and really you need that input to help you submit the best grant that you can. So if you have something like that, you really should be reaching out to our START team and picking up one of their pamphlets out there. We also do a lot of training and education. We have courses in regenerative medicine. We're training people in different parts. We have our fellowship program where we're trying to train the next generation of regenerative medicine, doctors. Just as a quick sort of overview of what we're doing at the clinical center, we right now have 39 active clinical trials. We've enrolled now almost 600 people. A lot of those people are some registry studies, some are more observational studies. But one of the things I wanted to highlight, which kind of will merge into what the questions I'm going to ask here, the panel, is that we started with a lot of oncology studies, and we still do oncology, but we've really moved into a lot more neurology studies. We have like 3 new ones in this first year: HIV cure study, we have AI study, we have a lot of different fields, and that's, I think that's one of the things that I've observed is the expansion of the field as we are moving to a more mature space with the CERM and the stem cell field. So what I'm going to do now is move over to just talking with our panel, and I wanted everybody to try and introduce themselves, because otherwise I'd be spending here all day just introducing all the accolades that all these faculty have. So why don't we start at the far end, and if you can just give me, introduce yourself and tell me what's the most thing you're most excited about right now in your Alpha Clinic.
This is I'm Mehrdad Abedi. I'm the director of Alpha Clinic at UC Davis. I'm a hematopoietic stem cell transplanter and a CAR T cell person. Our work is, just at this point, the applicability of the CAR T cells in many, many different disorders. From infectious disease, we have the HIV, cure study similar to UC San Diego, and working with UCSF on that. And we have multiple autoimmune disorder CAR T cells we're working on, to infectious disease, to different tumors, targeting those. So. And then the stem cell gene therapy, several trials that we are developing right now for different metabolic disorders, very similar to the previous talks there. It's a very exciting time for everybody now. Mm-hmm.
My name is Leo Wang. I'm a pediatric oncologist. I'm currently the director of the City of Hope Alpha Clinic. Our work really grows out of adoptive cellular therapies, initially for cancers. City of Hope is a very, very large cancer center. We also do some diabetes and HIV work. And so our work there has expanded beyond predominantly CAR T cells for cancer into definitive stem cell-based cures for all types of diseases, whether it's HIV in collaboration with UCSD, or the new FDA-approved gene therapies for sickle cell disease and thalassemia, or novel stem cell cures for diabetes.
afternoon. I'm Mark Walters from University of California, San Francisco, where I lead the Alpha Clinic there since its inception, 2017. I'm a pediatric hematologist, and my personal interests, research interests, are in hereditary hemoglobin disorders like sickle cell disease and thalassemia. The thing I'm actually most excited about is a partnership we've forged with one of the community centers of excellence. This one's located in Clovis, which is in Central California, and together there are some challenges to getting participation in clinical trials and access to approved therapies, cell and gene therapies, to folks closer to where they live, particularly if there are financial burdens to coming to a centralized medical center to get access to those treatments and participation in trials. So we're partnering to help develop good clinical practices for clinical trial performance in that location, but more importantly, embedding, we hope, we haven't been successful yet, but I think we will this year, trainees in that setting who would then stay there and be the ones to grow those programs locally. And what's great about this, and I think it's a real, I want to give kudos to CERM for thinking of this strategy. It's a way for us to build access to populations in our state that wouldn't ordinarily have access to these therapies.
Hello, I'm Catriona Jamieson. You've heard about what we've been doing very nicely from Sheldon Morris. I've been the Alpha Clinic director since 2015, since the inception of the Alpha Clinic idea that we would accelerate the development and implementation of stem cell related trials. And as you could hear over the last 2 days, we've expanded beyond gene therapies to cellular therapies to small molecules to biologics. And when it comes to cancer and preventing cancer relapse, we may need all of the above. So what I'm more excited about these days is working with Ludmila Alexandrov to really define who is likely to benefit from these very advanced therapies that you'll hear about from this particular group here. And when is it the right time to treat? So precision regenerative medicine. I'm really excited about our community advisory board. You'll hear from Andrew Shore at the end of the day, and why it matters for patients. When is the right time to intervene? That's where we can really reign supreme, I think, as a team here collectively. We work very closely with the Advanced Therapy Center, which is run by Dan Kaufman and Karen Chrisman, and you can talk to Holly about all the things that their team can develop. You heard from Innovative Medicine. So we can make the gene editing tools here, we can make the small molecules, we can make the biologics, but we need to collaborate, not just locally, nationally, but also internationally. So we look forward to expanding the reach of the CERM Alpha Clinics, and thanks to CERM.
I'm Mike Lewis. I'm the program director for the Alpha Clinic at Cedars-Sinai. What I'm passionate about is a condition called pulmonary arterial hypertension, which is a condition in which the small blood vessels throughout the lungs, less than 500 microns, are either obstructed or narrowed, and that eventually, puts a strain on the right heart, which becomes dysfunctional and fails. And the median survival, when there was no treatments available, which only became available in 1996, was 2.8 years from the time of diagnosis. In 2022, an analysis of survival in the treatment era with combination therapies, the survival increased only to 6.2 years, so still not very good, and there's no cure. So we were involved in, I was involved in animal preclinical studies, IND-enabling studies, which were very successful. This led to a phase 1 study in which we used cardiosphere-derived cells that are progenitor cells from the heart, but work by release of exosomes, and they're very bioactive cargoes of microRNAs, long non-coding RNAs, proteins such as IGF-1 and VEGF, for example, and they can work locally on any tissue or cells or systemically. And the phase 1 study, we just gave one dose of 100 million cells to patients with Ph and followed them over a year. Bottom line, it was very, very successful in terms of safety, which was the purpose of a phase 1 study, but also in exploratory efficacy. We had, actually statistically significant results in multiple different domains that we looked at in the exploratory efficacy domain. And that is now leading to a phase 2 study that I'm pleased to say we're involving about 21 different centers, but I've involved all the Alpha Clinic centers because they all have very excellent, pulmonary hypertension programs. The other thing is that based on our work giving these very same cells in now advanced phase studies that have completed phase 3 to patients with Duchenne muscular dystrophy with excellent safety in over 700 infusions and excellent efficacy in terms of handling the cardiomyopathy associated with Duchenne muscular dystrophy and limb muscle strength, they gave it on a repeated basis. And so we're going to be in the phase 2 study doing repeated peripheral infusions of these cells every 6 weeks. And, we think that it's likely to be highly effective. And the phase 1 study was done in the face of double or triple background Ph therapy. And the same thing will happen in the phase 2 study. So we're very, very excited about that.
Daniela Bota, and I'm the director of UC Irvine Alpha Clinic. Together here, we are celebrating 10 years of Alpha Clinic, at least at a few of our locations. And one of the most amazing things that we do in the Alpha Clinics is to collaborate together between the Alpha Clinics, but as well with our own researchers and collaborators. I'm a neurologist by training, doing neuro-oncology. It is quite amazing to see how neuroscience is getting changed. So from the times that we are trying to do CAR-Ts for cancer, and we're still trying, to now when we are seeing amazing results on all the inflammatory conditions in neurology, to the fact that we have gene therapies and a lot of the neurological diseases are monogenic diseases. So gene therapy now changes and transforms the life of those patients. So that's quite amazing. 10 years also mean with the CERM help that we all have the resources that we didn't have before. You heard about inpatient and outpatient facilities. We finally at UCI got our inpatient hospital unit in our new beautiful hospital near the campus, which is going to be dedicated to cell and gene therapy. We're also looking at something quite unique, and I'm looking forward to hear from everybody, the requirement from the FDA to follow the patients treated up to 15 years. And I cannot even imagine, because some of those patients used to live months, and now we are thinking about how we're going to follow them for 15 years. So I don't want to repeat something else that was already said, but really sky's the limit, and it's such an exciting time in cell and gene therapy.
Thank you so much. See all the exciting things that are done. And now we have a special guest too, who is one of our participants actually. So I'm going to call Tara Ghiglieri up, and I just got her bio. She's a former City Rocket Broadway performer. I didn't know any of this stuff till I saw this. I think we just want to hear about all this, all your life. But she was, so she's been from Texas. She's been in many movies, television shows, and commercials, and now is in San Diego. And we had a study called From Genesance, which was gene therapy for osteoarthritis of the knee. So she's going to tell us about her experience and give us a little bit about what the impact is of some of the studies we're doing. So Tara, do you want to come up and...
This is really exciting for me because I kind of geek out on this stuff. I always wanted to be a doctor, but I went the other route, right? So my name is Tara. As he said, I am a former Radio City Rockette, and I'm excited to share my experience with you today. As a lifelong dancer, I have a really high maintenance left knee that never really liked me very much. So all the years of dancing professionally for 35 over my lifetime meant a lot of abuse on my knees. Jetés and high kicks and tapping on, believe it or not, metal floors left my knee kind of like the diva of my body, right? Every step that I took, every stair that I climbed, everything that I did, my knee was on my mind. We should never think of our knees, right? But I did. That was my life. About 2 years ago, we moved to San Diego from LA, and it turned out to be great timing for this particular study. I was trying to figure out where the best taco joint was, and on Instagram popped up the Genomic study, which is kind of crazy considering that you normally get things like, fun recipes and med spas. So when it said, do you want to be a study for your knee? I said, yes. Yes, of course I do. Anything to help take my brain off of my knee 24/7. That's when I came across it, and I applied, and I spent a lot of time hoping that I would be a right candidate for it, and luckily I was. What really got me excited, though, was knowing that this was in its, I don't remember, stage 5, maybe, of the trial, and I knew that I was going to be getting the treatment. It wasn't going to be a double-blind study where I was going to walk out with a bum knee questioning whether or not I was actually getting a treatment. From the first day that I came in, everybody was amazing. Dr. Bota is just wonderful. I love her. Twee, who's been with me through the whole study, she's so efficient, so thorough, always making sure that I'm where I need to be and what's happening needs to be happening. They tried to make the whole thing exciting for me because, like I said, I kind of geek out on this. So I asked a million questions. What is it doing? What is it for? What am I going to experience? And they were really, everybody, Dr. Bukata, Twee, everybody was really patient with me and answered all of my questions always. Even on my birthday, when I had to come in for a follow-up visit, I got a birthday card, which was kind of next level for a study. And now I'm not going to lie, though, right? It was a difficult path for me. The beginning, there was a lot of swelling, a lot of pain, a lot of icing and elevating. But at one point, I even think that I questioned if I had done the right thing because it was such a journey for me for this gene therapy. But around the 3-month mark, something changed. I realized that I had not thought about my knee in a long time, that I could go for a walk or a hike or even do a dance class and not have my knee swell to the size of a watermelon after doing just a little bit of exercise. Now I can actually go for a dance class, go for a hike, take long walks with my kids and not be concerned that I'm going to be paying for the price, for the next week. I say how grateful I am for Genesance and for UCSD and for the treatment that I got and the ability that I had to actually change something that when I was 35 was told, you're probably going to need a double knee replacement in 10 years. And I feel like I am many, many years from ever having to need a double knee replacement, fingers crossed.
Well, there you go. That sounds, I'm so glad we were doing things. Who would have thought we would be doing, treatment of osteoarthritis? And yet, originally that was kind of the thing, the mesenchymal stem cell thing. And I don't know, I haven't seen one of those trials for a long time because they really haven't proven to be that much. So what I'd like to ask the panel is where do they see the future going? I think you've already kind of alluded to some of that, but I don't know if there's something more in particular that you're thinking, where's the field going? Like, where are we going to be doing in 5 years, 10 years at our alpha clinics? So I don't know where to start, but we can start at the other end again if it's or anybody that has a burning answer to that question.
I sat here not to be the first one, but anyway. Yeah, I think it was mentioned the sky is the limit. I think the field is just going to keep expanding. The beauty of this is that there's not going to be one treatment curing, but it's going to be just improving. New products keep going to come. We're talking about the CAR T cells, the first generation, second generation, third generation, just keep going. And the indication is going to expand to almost every disease that you can think of. For most of the medicine, I'm a physician. For most of the things, we have been doing management. We have never think about cure. We never cure hypertension, never cure, let's just say heart attack. We always manage those things. But this is, for the first time, is giving us opportunity to think about cure for many, many different diseases. And I think that's where the excitement comes from.
I think I want to just take a second to address the the overall climate. I come to this symposium every year and just am floored by how amazing it is. And Kat and her team pulled together some of the just, trailblazers and brightest minds in the world. And I was privileged enough to sit with a few of them yesterday on the shuttle ride back to the hotel. And they were talking about how we are currently in this environment of shrinking, of pulling back, and frankly, a little bit of fear, and how people are retrenching and maybe downsizing, and that's absolutely the wrong strategy. And one of them said, in order to succeed, you need to sprint straight at that brick wall, full of the confidence and the faith that you will burst through it. And at first I was like, well, what if I'm a muggle and I just hit that wall? And so that, that was actually a question for me about survivorship bias, which is this thing where if you only look at the people who make it through, you ignore all the people who don't, maybe you start to think that that's the way that things have to happen. And that's maybe incorrect. But the more I thought about it, the more I realized that actually, that ethos is not about hubris. It's actually about being bold and also understanding that sacrifices are important. And we really have to be in a situation where we need to continue to sprint straight at that wall, because otherwise you're not going to be able to break through it. And more importantly, I think if we stop sprinting, people will see that we're not sprinting and they'll stop sprinting. And then eventually it won't matter whether the wall's made of brick or even just imaginary. We'll be penned in by these walls that we're not willing to challenge. And so I think the value of this network and of CIRM and the work that we all do is that we are the standard bearers for this sprint at brick walls, and we cannot slow down.
I don't know how I'm supposed to follow that. Better. So I was amazed by Stephanie Cherky's work in Friedreich's ataxia, and just a couple of points to make about that work. First of all, the CRISPR technology came out of basic translational research, was discovered in a basic lab. Then I think the Nobel Prize was awarded because people like Jennifer Doudna realized that this could be a research tool and a tool for therapeutics. And so, a little more than a decade later, I'm using it to correct the sickle allele in a gene therapy trial targeting the hematopoietic stem cell. You heard about Stephanie's work. Sickle cell disease is the least rare of the rare disorders. Friedreich's ataxia is very rare. And frankly, there's really no commercial model to develop these rare orphan diseases. And so it's extremely important that CIRM and institutions like CIRM support this work, because otherwise, the whole goal of precision medicine, of the genomics work, this platform, is to apply it in a therapeutic way, and we won't be able to do that if we don't have the resources. So I'm really excited about the work that we're doing, that Stephanie's doing. Fyodor Urnov at the Innovative Genomics Institute, which is attached to our Alpha Clinic, is developing a platform whereby he's targeting, I think, 7 of the hereditary causes of hemophagocytic lymphohistiocytosis, which is this terrible disorder in children that's an inflammatory disorder that has to be treated by a bone marrow transplant. So using gene editing to prevent that disease would be spectacular, but also a rare disorder. So am I addressing your question? Okay, good. I'll stop there.
Thanks so much. So I think what we're all doing together, you see from the 6 of us, and there are 9 Alpha Clinic directors in the state, we play to win. So if you go to the racetrack there over in Del Mar, they say win, play, show, or play to win. Only play to win. What's the alternative? So if you think of us, we're your Marines. So David Beck here is a Marine. We have other Marines in the audience. we don't see the wall, together we'll run through anything, because the luxury is to give up for patients who can't afford the luxury of us giving up or even slowing down. So I agree wholeheartedly with you, Leo. I think what's changed is we have innovative technologies, and we heard from our Rocket here that not everything's inevitable, actually. And that's the part that I was alluding to with Ludmilla and others in the audience that make these predictive tools. We know when is the right time to intervene early. We don't wait for really advanced disease. So in your case with the mesenchymal stromal cell trial, mesenchymal stromal cells are little anti-inflammatory factories, but on their own, they're usually not enough when you do the gene therapy on those to make the IL-1 receptor alpha trap. So it sucks up all the IL-1, the inflammatory cytokine, and the synovium. We can see the way you're walking that was a game changer. So that's what we want to do again and again and again as a team of Alpha Clinic directors, where we can do accelerated confidentiality agreements, clinical trial agreements, registry studies, so we understand the gap, what is the need in the community and how do we intervene as quickly as possible. What we want is not to allow tissue degeneration to occur in the first place, and we really want to sprint to the finish line so that people never face those draconian therapies. I'm a recovering bone marrow transplanter. I think I need a 12-step program for that. And now you can see our therapies are becoming much more bespoke and probably more effective. And, Stephanie gave a great talk about gene correction, gene editing. There have been Nobel Prizes. But I would say the most important prize are patients, and not just extension of life, but improvement of quality of life. Health span, as Lee Stein always tells me, is the most important arbiter of whether we're getting it right or not. So I'm thrilled to be able to work with our team here. I just wanted to follow up on a point here. We're all bringing substantial resources from our own institutions. We actually had to have a one-to-one match, but when you can see the talent in the room, it's a combinator. It's a much more than a one-to-one match. It's a multiplier. And when you bring in our patients who are forces to be reckoned with individually and together, it's a little scary. No, it's fantastic. Thank you for supporting us. We can do more than we expected. Thank you.
Well, I think that regenerative medicine and the Alpha clinics are going to go from strength to strength. I do applaud, all the studies that are being done in rare conditions, because I think that helps to advance technologies and understanding. But I do think we need to also consider common conditions. And, at Cedars, Eduardo Marban for years has been looking at, common cardiac conditions. And one of the things that they're concentrating on now is called heart failure with preserved ejection fraction. That accounts for over 50% of all heart failure that there is in the country and in the world. And, so I think we do also need to extend these technologies to the more common conditions, because that's going to provide way more bang for the buck in the end run. But we need to do both. We need to do the unusual and the novel conditions, but we also need to tackle all the common scourges that, plague us in the field of medicine.
So it's very interesting, because I was thinking something very similar. We are talking about rare conditions, and they are severe, dramatic conditions. But in the end, as physicians, the causes of that are still, unfortunately, remaining the same. And I'm going to talk about part of what was CERM intention and CERM funding, which was focusing on neurologic disorders. And for people that are old as me or even older, you remember that there was a decade of the brain. And there was a decade of the brain in the 90s. And the reason for that was, between many other things, trying to find a cure for Alzheimer, trying to find a cure for neurodegeneration. It's wonderful to see the clinical trials in Parkinson moving ahead. We still don't have something effective for Alzheimer, which remains one of the major causes of disability. we still do not have cures for many of the cancers that are still killing a big number of people. And I think that in the next 5 years, that's going to be where we're going to find ourselves, hopefully in clinical trials, if not in effective treatments and cures for those large, very important conditions, because in the meantime, we're going to be curing all the rare disorders. So then we all can concentrate on the big ones.
Thank you. Yeah, and that was kind of like you were answering some of the other questions I had, which is about, that tension between, focusing on rare diseases and common diseases. And I think the answer is we should be doing both, and we hope that mandate at CIRM continues to be both, that we can serve it all. We need wins, and we need to continue to focus on ones that are going to be successes to show, because otherwise, people say, well, we gave all this money for the in the prop, and what have I got for it? So we have to show results for people. One of the other things in the prop was really this idea of community centers of excellence. We heard a little bit about that, and so I wanted to ask the panel about how we're doing with that. What are we seeing movement in community centers starting to be able to maybe do some of the work in regenerative medicine? What are going to be the barriers to see that happen? I mean, this is something that the prop actually wanted us to do, but I want to know what the progress has been. Why don't we start? We'll start at this end this time.
It's going to be hard. We are seeing progress. So UCI works together and very collaborative with UC Riverside. And they are, by all means, our partner in the grant and a future center of excellence. But when you think about doing the clinical trials, the question becomes which part of the clinical trials. And I think the great role for many of those centers is for patient education, patient recruitment, community education, because when you go in Inland Empire, and I know many of you do, you find different kind of information and knowledge than what you see around our big academic centers in our big cities. So will we do anytime soon cellular therapies in patient transplants in community hospitals? I don't think so. But could the other outpatient treatments be administered? I think they can. And can patients be followed up before and after the intervention in their hometown, in their home clinics? Absolutely they can.
So I think that the community care center concept is a good one, but I think we also need to be realistic about what's feasible and what isn't feasible. And, just talking for myself in the pulmonary hypertension world, I mean, most cardiologists and most pulmonologists who deal with pulmonary hypertension, who deal with pulmonology and cardiology, know very, very little about pulmonary hypertension management and excellence. And so we get sent lots of patients from these folk where, they're just not treated well. So you do need centers of excellence and expertise to deal with very, very complex conditions. And so I think we have to be realistic about what could be accomplished in a community care center, in a community hospital setting, and, what really needs, kind of higher level of care expertise. But I think whatever can be accomplished in that setting, I think is a plus and an advance.
I think when I remember the Prop 14 language, community care centers of excellence in what was my question. And I think they're great at being able to communicate what we're trying to do in terms of applying the most advanced therapies for people with the most complex manifestations of rare diseases and common diseases. And that's where it can be a funnel to come into centers of advanced care. I think for precision diagnostics, they're perfectly well suited to be able to do this. And this is what has changed in the last few years, actually 10 years, since we started the Alpha Clinics. You couldn't do molecular diagnostics at a local community clinic. Now you can, because of everything that we all went through with COVID. So we know that those diagnostics can be applied locally, and that is going to be a game changer because we can't just see 1/10 breadth of the patients. We need to see all the patients. And I would say the community care centers of excellence establish a very important framework for thinking about rural health and how we apply things beyond the coasts of this country and in other countries like India, where you have really stellar care in these centers, and it's the same in the US, but not so stellar care in the middle. And, we'll look to define excellence because on grants, you have to be outstanding. So maybe that's the difference with the alpha clinics. But I think the difference is we need patients to know we're here for them, and we need patients to be involved. And I think the community care centers of excellence bring patients to the table.
Yeah, I don't have very much to add except that based on what I've observed and my personal experience working in a safety net hospital for a while, these centers are resilient and they're resourceful, and they want to deliver the full spectrum of care for their patients in their community, and they are ambitious. They're getting accreditations to deliver cellular therapies, gene therapies in their location after they've been approved. And so we should probably get out of their way and give them the resources to do what they think they can do. And they need a partner. That's really clear. So I'm agreeing with that. And they have to deliver excellent care. But they're committed to that. So that was just the additional point I wanted to make, is that this is a real resource and an important advance for our state to deliver these cares in the community.
I totally agree with that. And it's even more than getting out of the way. I think it's enabling this. I mean, they are ambitious. They're doing it. These therapies are being democratized as we speak. Texas just passed a law saying that you can get CAR T-cell therapy without requiring FACT accreditation, which is currently an organization that's helps to sort of get these therapies paid for. And so that was a real barrier to getting these therapies in like Lubbock. So Texas passed this law that now you don't have to do that. And the Foundation for the Accreditation of Cellular Therapies is working on developing new standards so that community centers can develop these therapies safely and effectively. So it's definitely happening. And I think where we have a real opportunity is to lead the way and model how to safely and effectively enable the dissemination of these new technologies.
I completely agree. One thing I want to say, some of these treatment right now seems very sophisticated and cumbersome to do and complicated. But as the technology improves, as the knowledge improves, I think we can easily do many of them in the community setting there. We are working, for example, with Fresno, as Mark is working with them, as well as with Loma Linda, to actually do not only administer CAR T cells locally, but actually manufacture CAR T cells locally, because we can automate many of these things there. Right now, we do apheresis for collecting CAR T cells. Actually, we're working on just doing a blood draw and manufacturing CAR T cells. That everybody can do that, and then automating everything, so you don't need a very complicated GMP facility, and they can locally make these CAR T cells and administer those. And as the technology improves with the CAR T cells, the complications improve. So at the very beginning, when we did the CLL, CAR T cells or AL CAR T cells, there were so much side effect there. Everybody was going to ICU. People are so afraid of that. There was a very extensive REMS program. But now as the technology improves, the vectors improve, the toxicities are substantially going lower. And, we don't need many of those safeguards that we used to do. And we think we can not only administer those in a community centers, but even manufacture them. The problem is that, originally it was a very rare disease. ALL, it's a very rare disease. Fortunately, it's a bad disease, but it's a very rare disease. But as these technologies getting into much more common cancers, there's no way the academic centers can keep up with doing these things. If you talk about tens of thousands of patients requiring these now, we can't handle those patients, in academic centers there. So there is no way we can do this successfully unless we can, expand to the community centers there and everybody be able to do this.
Very informative. So the other thought I had is, well, how could we be better? How could we better as alpha clinics? What things would you need to improve the structural changes, resources, considering the changes that are coming, what other kinds of disease targets we're having. So again, I'll start with Danielle. Is there anything that you think we could be doing better in terms of into your site and working with other sites?
I think that the most important part on which we can do better is to be more and more effective with our resources. And that starts from, we have very strong clinical trial operations, but having things automated, being able to use artificial intelligence and machine learning to optimize the way on our researchers work. The days that you go on the chart and you start writing the same record 3 times and getting it on paper and getting it in the EDC, those things have to be resolved, and we can resolve them together. We are all involved in levels of optimizing operations. I think the other thing that we can do together, and we have done it all through, but there is always better, is to learn from each other and to really function as a network. The last but not the least, one of the things that we all thought about, but we have yet to put together, it's a certification system for cellular and gene therapy for people that are not transplanters. Because right now, talking about the FACT accreditation, talking about the work that Leo is doing, there is a need to democratize those therapies, not only to centers, but also to different branches of medicine.
Yeah, I think that, first of all, I love the alpha clinics, and I love them for the reason of collaboration, which is just beyond, what I could have even imagined. But I think there's room for even more collaboration. All of the alpha clinics have special offerings, which I think could be more utilized. I'll just say, for example, AI is one where it's just advancing at such a rapid rate that, it definitely could be utilized, and, all the alpha clinics could weigh in and be part of that. So I think, and so that's one thing that could improve. I think the other thing is that I think the alpha clinic program directors and other personnel, program managers, maybe could be, also more involved in the changes that, CIRM put about, rather than us learning about it, towards the end and then having to, kind of, in crisis mode, contact, board members and, people at leadership positions and say, this doesn't seem to work well for us. It's not going to advance our needs and the ability to do clinical trials.
Yeah, I was thinking, I attend on our inpatient cell and regenerative medicine service, and it gets pretty complicated now that we're expanding CAR T cells beyond ALL and other heme malignancies to actually solid tumors and autoimmune diseases, as Danielle is pioneering, and others. We still need tocilizumab or a biosimilar stockpiled. This is not in a standard pharmacy. So I think if we're going to have community care centers be involved in the distributive manufacture, which makes so much sense, and the administration of these cellular products, they have to be able to have these therapies on their pharmacy books, where you can just dispense them. We had to stockpile it at a time where people were using Toci off-label for the side effects of COVID. So we still have challenges. I would say the biggest challenge is cost, and that's the cost to patients. So I was a bit shocked and appalled, actually, where I had to do a Medicare denial response to a judge for an FDA-approved therapy. And that was quite shocking to me. Like Mark, we're in the UC system, like Mehrdad, like Daniela. We have a lot of Medicare, Medi-Cal patients. I've never had a Medicare denial where I had to go to a judge. And the patient said, "You're not much of a lawyer." I said, "Thank you." And we won the appeal, but this was an FDA-approved therapy where Medicare thought we should have given the combination to begin with, and was second guessing the judgment of the care team in terms of how to sequence in. the chemotherapy. So I think we have to look at bundled payments. There is a solution. We have that for bone marrow transplantation for all of us who trained in that. And these therapies will likely live in academic and community care centers of excellence as we really expand the infrastructure for paying for the drugs that go with these therapies, because it's therapies alone aren't enough. You have to give these other therapies that will prevent cytokine release syndrome and ICANS and all these other things that go with them. So that's what I would say. We take on the bundled care payment model. Sometimes, and this is taking Maria's term, there's innovation to commercialization, but this is commercialization, or this is doing the opposite. This is internalization of what had been commercialized or could be commercialized. And I know that doesn't always fit the pharma model, but it fits the patient model. And if we're all going to be patients someday, and we are, then what would we want? I would want a bundled payment plan where I know exactly what it's going to cost to get Stephanie's gene therapy for a common or a rare disease, to get the cellular therapy that I need, or the small molecule, or the biologic, or the combo. You know, how many people here want to cure cancer?
Okay.
You know, if we want to wipe cancer off the planet, we're going to have to start earlier, but also have this bundled payment approach where it's irrevocable and never, ever revocable. So the irrevocable trust idea is what we have to have as physicians and scientists, and not just as a kind of aspirational thing. So I know those are strong words, but that's what we're facing right now, I think, is our biggest challenge, is the cost and how to overcome the cost of care for patients.
UCSF, which is an enormous place, more than 30,000 employees, just professional graduate level training. So getting the word out amongst this ecosystem that there's an Alpha Clinic to help teams has been challenging, and I think we're making progress, but it isn't perfect yet. So we continue to work on that. And then related to that, particularly with the manufacturing infrastructure that was supported by CERM, we developed a very nice early phase clinical development team. And so we are actually spending almost as much time at early phase preclinical development support as in addition to our clinical trial support. So the Alpha Clinic scope of services and skills has expanded, and I think that's really important, because the science is exploding, and more and more basic labs are coming up with ideas that look really enticing for human therapeutic development. So and we need to be able to support that. So those are the two big challenges that we're facing.
Yeah, I have a lot to say on this topic, but a lot of it's been said already, and I know there's like audience questions and stuff. So maybe, Mehrdad, you can.
The only thing I can add is that, between all of our Alpha Clinic and also our Community Center of Excellence, we're covering all California. That's more than 50 million people. I don't know what was the last population of California, but more than 55 million people. And we are in a very, very unique situation when it's better than any collaborative group in the whole US, that we have so much power, we have so much expertise between our groups there that can make really meaningful changes in cell and gene therapy. And I really want to use that leverage to come with common strategies between all of our groups there, pick up maybe few really life-changing, maybe for common disease, and work together as a California-only, Alpha Clinic-only clinical trials, and elevate that and show to the people of California and the whole country that we are able to do it. Again, nobody else in the US have at least that capacity to do this kind of transforming work.
Okay, we still have, I think, about 20 minutes. So I think what we can do is take some questions from the audience.
So there are some new technologies now. For example, stem cells can be lyophilized and stored and made off the shelf. And there is also another technology, I don't remember it off the top of my head, but there's a company that will allow you to store cells at room temperature, not through lyophilization. It's a different preparation. So this could be useful in the increasing production of stem cells, even CAR T cells, although those have to be tailored, of course, to the individual. And are you, I guess you have a stem cell production facility with each Alpha Clinic. So are you considering adopting this kind of technology, like lyophilization or other ways to prepare stem cells and store them and ship them easily at room temperature?
I can take that. I think for stem cells, they're unlikely to be functional after lyophilization because they rely on gene expression pathways, stem cell pathways. You can lyophilize platelets, though, because they're anuclear. So that's an opportunity. But so far, stem cell technologies rely on having living cells, and I haven't seen technologies that preserve that function of regenerative capacity that we need the stem cells for. If we just think of stem cells as factories like mesenchymal stromal cells, that's a little different, but those aren't inherently stem cell populations because they can't self-renew or, in other words, regenerate.
No, this must can keep cells alive and also exosomes.
Yeah, I mean, I think that it really depends on what cell you're talking about, whether it's a true stem cell, whether it's a progenitor cell, whatever it is. With regard to some of the kind of progenitor cells, like the ones that I work with, they can be frozen. They can be prepared and frozen and, at -80 and be available to use and, defrost and use when needed. So, that kind of, assists in that regard. But I think it really depends on what product you're talking about and the circumstances around that production.
I don't have any questions. I just want to, it's kind of like a testimony, appreciation. First of all, I want to say thank you for Dr. Jamieson for allowing me to come here yesterday and speak, be a part of this great event. I've never been a part of an event like this to talk about how important something that's very personal to me and how I can be a better advocate for. And the reason I say that, and I appreciate her saying stem cell donor. I'm also a bone marrow donor. Platelets and plasma. Since I was 17 years old, I've donated my first pint of blood. I was 17. I lied that I was 18. And former military, also worked at the DOD and I just retired from DOD. I found out in May that I had stage 1 breast cancer, and I had surgery in August. And City of Hope has been very, very, very nice and very professional and helped me through the process. So I had a couple of tears when you guys were talking because I lost a brother from 4 different types of cancer 2 months ago. And I also lost a classmate 2 years ago from sickle cell. And I think it's important that we keep fighting, right? And some people's walls may not be as big as others, and how they run might not be as fast as others, because my knees are bad too, just like she said her knees. I have bad knees from the military, but I think it's important we're here today. And what we can do today and what we can do together really makes a difference, because just being here around all these very highly educated people who really can, like you said yesterday, spent 25, 35, 40 years of your life to do what you do to support and save lives really means a lot to me. And I want to tell you, I'm going to start crying. But it's been a very challenging year, and I'm really thankful for the opportunity to be here, to be supportive, and also be an advocate to one day, hopefully I can donate money back into these institutes to help with this work that I do at the end of my life. So thank you so much.
Thank you so much. I have an online question from the online audience. What are the next low-hanging fruits for cell and gene therapy translation and commercialization?
I've got to sing Mehrdad's praises here. I think distributive manufacturing. I think getting the manufacturing of the cells and the gene therapies to the right places, the rural communities, and everything that Mark has led in terms of the gene therapy initiatives, making sure that they get to people with sickle cell anemia. What Leo's doing at City of Hope, conquering cancer. No pressure, Leo. What we're doing here with everything to do with pulmonary hypertension, what Daniela does for people with brain tumors and for autoimmune diseases, neurodegenerative disorders. I think the low-hanging fruit is letting this team work together to bring things forward together and run through those, apparitional walls that are mirages, I would say, Leo. I think we can do this, and we just need support, financial too. Yes, please. Thank you, David. If there's a crowdsourcing opportunity, yes. We have our teams here. If you want to raise your hands from our Stanford Stem Cell Clinical Center team, we have a START team, Matteo, Peter, Fabiana, Kendall, Colin. We have our whole team here, Tuya in the back. We have Kenson Lamb there as one of our physicians who attends on the service. We have Dan Kaufman here. So we have our whole team, Jameson here. I call him the other Jameson, but he's a great Jameson. Anyway, so we have teams here that can do this together. I think that's the low-hanging fruit because we each have teams like this. That's the multiplier effect.
So maybe I can add something different and related at the same time. We do a lot of treatment. I don't think we yet have universal diagnosis. So when you hear all of those wonderful stories about all those rare disorders, always my mind goes to all the children that are not diagnosed and how much of a difference things made when we started to do neonate diagnostic, and which right now sequencing being at such a low cost, it's nominal. Being able to spend that $100 on each neonate so we know as they are young and they can be repaired and they can be helped, who those kids are and getting them to the right care, I think that's going to be something of a low-hanging fruit, needs investment, but also something amazing that will help all of us help our patients.
I have to say that I was really impressed by two Intellia-sponsored trials. I mean, it's just a lipid nanoparticle decorated with ApoE, targeted the liver, and it delivers a guide RNA and the nuclease, the Cas9 nuclease, and it cured transthyretin amyloidosis. And using the same platform, the only thing that changed was the guide RNA, hereditary angioedema. So these are like liver targets for defective enzymes or proteins. And there's got to be gobs of them, So I think that's a low-hanging fruit, and I bet we'll hear a lot more about it. Really simple design, platform approach, and using the gene editing technology that's so efficient, remarkably efficient.
I think the low-hanging fruit right now is bringing the cost down because you don't need that much to do that. We're already doing that. We already doing. The cost of a CAR T cells, if you want to do CAR T cells right now, commercial, just the product itself is $500,000. And the sickle cell treatment is $3 million. And we can do it in academic centers. We can do it in Alpha Clinic in a fraction of cost, 1/10 of the cost there right now. If you have resources that, from CERM, from community to be able to do that, we can do this much cheaper, so everybody can benefit from that. Because again, right now we're talking about rare diseases, but as these technologies expand to much more common diseases, then there is no way that we can afford this. And bring the cost, I think, is critical.
Other questions from the audience? Got a couple, 2, 3.
So currently with most recent versions of CAR T cells, the patients, do they still need the EMG therapy after the CAR T cell procedure is completed?
Yeah, if you're targeting B cells, about 10 to 20% of them may need IVIG. But again, that's only for certain type of CAR T cells that targeting certain disease. Many, other CAR T cells, they don't need that. Depends on what the target is. Each CAR T cell has its own side effects. And the management is, different for each, target. But again, down the road, many of these things, I think, will be less and less as far as side effect, as far as toxicity profile will be easier and easier.
Fang Chen from Biotherapeutics, a microRNA therapeutics company based in Pennsylvania. My question to the panel: have you tried to expand the some of our clinics beyond the border of California. And then that's the first question. The second question is, have you tried to tap into the federal government laboratory manufacturing plant that can help you manufacturing the FNLCR, I believe, that can manufacturing for anything CAR-T for cancer or even rare diseases now?
I think what we heard is there are different ways to make CAR T cells. There are in vivo CAR T cell strategies with lipid nanoparticles. You saw 2 acquisitions here in San Diego, Capstan Therapeutics by AbbVie, and more recently Orbital Therapeutics, which is one of Gene Yo's companies, by BMS. So you can have the lipid nanoparticle technology that delivers the modified RNA that will allow you to make the CAR T cell in situ. So I think there are different ways to make these products, and it's not just T cells. Dan will tell you about NK cells and other cell types. I think that we're moving beyond T cells to NK cells to back to stem cells. So I think there are different ways to manufacture, and certainly we're open to other people, not just in the country, but globally. And, we just need a California PI, the way CERM rules are written, but we're certainly willing to expand. I would also say we're working on making these CAR T cell therapies more effective. So if you think of what happens in the tumor microenvironment, interferon is repressed by molecules like ADAR1, and we've made a drug that blocks that. And there are many strategies to improve on CAR T cell and any adaptive immune effector therapy. So I think there are opportunities for manufacturing collaborations, and I think that's where we can get a bit more savvy in terms of these industry academia collaborations as low-hanging fruit, as Aaron was alluding to.
I think CERM has been open to partnerships with the NIH. I know for a fact that they have entered into a memorandum of understanding for a particular indication or particular focus of research. So there is a precedent for that too.
Yeah, the Frederick Laboratory is actually a federal owned facility. They can manufacture this stuff for you almost at in-kind service. So I think you should tap into that. It's actually for clinical trial for cancers as well as rare diseases that can do that for you.
We are working with NIH in different projects for sickle cell as well as for triple anti-lymphoma CAR T cell, and they are manufacturing. They're going to start manufacturing their own stuff locally there. And the idea is that eventually it's going to be, manufactured, widely, as well as even for, countries outside. They're going to find a process that we can do it, much, much cheaper. So in Africa, they can do it themselves, and they don't have to, they don't have to rely on a big company making it for millions of dollars.
Right. So I want to thank all of you for running the Alpha Clinics and for working with CERM. The way you guys are reaching out to community care centers in rural parts of the state, I think is a really incredible initiative. I was curious though, is the expectation for these community care centers to have like full manufacturing capabilities themselves, or is it like sending cryopreserved final product from maybe an Alpha Clinic manufacturing site or something like that? Is that more the idea of, or are these community care centers going to have the full manufacturing capabilities to assist the community?
For the common diseases, if we expand those to the common diseases, yes, we are hoping that we can do this. Again, if you want to try to treat the lung cancer with CAR T cells, or lymphomas, yes, that's doable. When you're coming with the rare diseases, I don't think that's financially and logistically reasonable to do that. I think academic centers still can take the lead on that one. And depends also how complicated the process is. For stem cell gene therapies, it's complicated, and I think still at the end of the day, the academic centers will do that. But for some other, low-hanging fruit there, like I think CAR T cells, we can do it probably locally.
So maybe I can add to that, because the Community Care Centers of Excellence is a designation that's coming with a grant, or will come with a grant more correctly. And a requirement is that they are able to treat patients, at least majority of them. building a CGMP, not just having a clean lab or you have a closed system to produce those common CAR-Ts, it's a very expensive endeavor. It's tens of millions expensive endeavor. And you cannot close and reopen because you need to constantly keep the space working and the standards to make human products. And in the way that the medical system is working right now and all the challenges that we have, probably it's going to make sense only for very few places, if not no places, to have this kind of facility.
Yeah, I think it's a staged approach. So current state, all of the FDA-approved CAR T-cell therapies are manufactured by the companies and then shipped to the site for delivery. For the clinical trials that we're all talking about, most of us manufacture them, at an academic site. But as we talk about a lot of these improvements, we're thinking about simplifying manufacturing, shortening manufacturing time, decreasing cost and complexity. So in a future state, I think it is conceivable that a lot of these places might be doing point-of-care type of manufacturing and/or delivery. Right now, it's more about transferring that expertise, to give them the training and the know-how so that when we get to that future state, they're ready.
Thanks. So there is a program of ARPA-H. John Shield is a program manager, and they have money to give away to, a lot. And then that's a good opportunity. And I think it's tax money. We all pay for it, and we should get it. But the other question that I have related, that was a comment, but I have a question related on what about so there is a small molecule that was discovered for pulmonary fibrosis. The principle of it was made in a collaboration in NIH, NHGRI, and NIAAA. And then this person ended up getting a molecule that is kind of a similar of a mu-opioid receptor agonist. And then it turns out that was really good for pulmonary fibrosis. But then we ended up finding that it also helps losing weight. And then Novo Nordisk bought it. And then we were interested in using it for Hermansky-Pudlak syndrome patients. And then we couldn't. And then they are reluctant to do that. Like, they don't want to start up. So do you have any avenues, or it's kind of a dumb question because I don't know anything. So I came to be instructed and taught. So what would you do? Do you know any avenue in which you can approach to these companies and say, like, hey, let's say that I pay this clinical trial, just give me the molecule, just provide me the molecule and let's sign whatever you need to sign, and then just let's benefit these patients. Because it's rare genetic disease, so it's less than 10,000 people. So,
well, there's right to try legislation. There's also compassionate use, and as Alpha Clinics, we can do that collectively. Actually, compassionate use INDs, and that's what I was alluding to before. It's not just about cellular therapeutics and gene therapy. We can do small molecules to enhance endogenous repair and regeneration. So I think right now we have the expansive capacity to move beyond cellular and gene therapies. And if you read Prop 14, it's about stem cell research and application of stem cell research to the clinic. And that's all indications: small molecules, biologics, cellular therapeutics, gene correction, gene editing. I think we have the full spectrum of activities. I think the hard part is looking after people once they've had complications for their diseases. So not everything is a side effect of the drug or the cellular therapy. It's just the diseases have their own side effects. So often with compassionate use, we say, hey, we'll get the drug, but nobody can pay for maintenance of disease control. And that's where we need to change how we think about that.
Yeah, the field is rapidly advancing, and I'll just give you one example. At Cedars, the cell that I mentioned, the cardiosphere-derived cells, they work through the release of exosomes. The exosomes are very, very biologically active cargoes, and we've mined the exosomes and come up with several, RNAs, particularly the Y RNA, although there are 3 that have shown marked biologic activity and can replicate the effects of either of exosomes or of the cells themselves. They've been manipulated so that they can be given even orally, or can be given subcutaneously or intravenously. So these, and we've done preclinical studies in this scleroderma mouse, for example, looking at marked effects on pulmonary fibrosis in that model. And I'm going to be doing a study in pulmonary arterial hypertension model. So, that's something new, innovative. And, as I say, things are just moving at such a pace that who knows what's going to, be available in 5 years' time.
Okay. Yeah, well, and I don't want to take too much more people's time, but you're going to be hearing in the next part a little bit about our biomanufacturing here. And I want to get back to this point about point of care manufacturing. And I want to agree wholeheartedly with what Daniela said. I, quite frankly, don't see the point of point of care manufacturing. Collecting the cells at a local site, community site, doing the apheresis makes a lot of sense. FedEx works really well, right? And, shipping the cells overnight to a specialized center that's regulated and approved makes a lot of sense, and more sense than building 20 of these centers around the state, all of them that need manufacturing license, all of them need regulatory teams, all of them need manufacturing teams. That's a very redundant effort for something that, even if it's common, if you're doing 100 lung cancer patients, I don't know, a month, that's not something that's going to happen. I don't really see why we want to build a bunch of redundant manufacturing centers. Maybe somebody can help me out. Also, CIRM, each one of you has a CIRM-supported biomanufacturing center. We need to be using these centers, not repeating this somewhere else.
Sounds like maybe you have a lot more available slots than we do. We have very long wait times.
Yes, absolutely.
So building capacity is super important. But even more important, I think, is not so much. I agree that we don't want to duplicate things. I think we're talking about miniaturizing things. So, maybe moving out of a GMP space into a GMP-like space with a closed system, which, Mehrdad can speak to this, but maybe I'll just turn it over to Mehrdad.
So we probably have one of the biggest GMP centers in California, academic center, and we are still booked. And again, we are talking about right now, in the biggest scope of the things that we're talking about, very, very few projects. When we get to, autoimmune disorders, you want to treat all of the autoimmune disorders. You want to treat all of the rare diseases. There's 7,000 rare diseases that we're going to treat there. We gotta treat lung cancer, breast cancer, colon cancer there. And I understand there are other technologies that may make it much easier. We may not need autologous cells. We may, your work also, different source the cells that we can use it for everybody. But if we stick with the autologous, that definitely there are some benefit from that. We're going to be very quickly overwhelmed. And again, we're not talking about building new GMP facilities. We're talking about
approved therapies. Approved therapies are not going to be done.
No, not approved therapies. No, yeah, but we're talking about we're actually working with FDA. We are working with FDA to make this a process that can be approved and have a BLA for it. So the idea is that very similar to a big company. If a big company has several centers there is a mother basically company there, and they have several manufacturing centers there, and the BLA is basically showing comparability of all the centers doing the same thing there. So you can do the same idea for a non-profit organization, and we are working with non-profit organization to be a BLA holder, and multiple groups there. They can do it off the shelf in a clean room in a closed space. They can do their own process very quickly and extremely cheap. And then the idea again, collecting the cells and giving fresh back to the patients immediately in a 2 or 3 day turnaround time. It's going to be very appealing for many of the diseases, at least for cancer diseases.
Thank you. I think we need to break. It's coffee break time. Thanks, everybody. Terrific panel.